Good afternoon, everyone, and welcome to another episode of the podcast. Today is episode 64, and I'll be talking about fungal reproduction. In pretty much every episode in the series before this, I've mentioned aspects of fungal physiology or pathology that involve reproduction. I've always touched briefly on the subject, but never gotten into too much detail. If you had always wanted to hear more about that aspect of fungal biology, then today is your day, because this episode is all about the ways that fungi replicate. And there are a lot of ways that fungi replicate. There's a lot of them. As a kingdom, fungi have a huge range of reproductive tactics, compared to animals and plants. They can reproduce asexually through fragmentation. They can reproduce asexually through the deliberate production of buds, or with spores. And they can reproduce sexually, either with gametes or through anastomosis and plasmogamy. In most cases, fungal reproduction is just weird and complex. I want to introduce this weird world of fungal reproduction with the group known as the fungi imperfecti, or the Deuteromycota. Although they aren't classified like this anymore, these fungi were once classified literally, taxonomically, as, quote, imperfect fungi, because they don't appear to have a complete reproductive cycle relative to other fungi. The imperfect fungi have only ever been seen to reproduce asexually through the use of spores. It seems as if they're largely stuck in what's called the anamorphic stage, which is a stage in the fungal life cycle where they reproduce vegetatively, asexually, as part of their growth through this portion of the life cycle. They never really seem to reach the teleomorph life cycle, where the fungus reproduces sexually and produces a fruiting body. For the Deuteromycota, this just never seems to happen. In contrast to the imperfect fungi are the so-called or the implied perfect fungi. But all that this means is that they faithfully go through the entire life cycle, with an anamorphic stage characterized by vegetative growth and asexual reproduction, and a teleomorphic stage characterized by sexual reproduction and the growth of a fruiting body. I'll go into more detail on the reproductive cycle and the fruiting bodies later in the episode. Because fungal reproduction methods are so varied, and they occur during various stages across the fungus's life cycle, there's going to be a lot of information to cover. So I'll be exploring each method of reproduction in as much detail as I reasonably can, with consideration of time constraints. I don't want to make this episode drag on forever. So with that said, it seems appropriate to start at the simplest, most basic method for fungal reproduction, which is an asexual process called fragmentation. It's the simplest method because it just involves the mycelia physically breaking apart to form two separate chunks of mycelia. What's pretty interesting is that fungi can survive fragmentation caused by physical damage, and they can also engage in a deliberate kind of fragmentation with their own chemical processes that's a lot safer. The mechanical damage is pretty straightforward. Imagine you have a mold, or a yeast colony, or a fungus, just any kind of mycelia that's just out there growing on a rock or a tree, just somewhere in nature. Now, if an animal comes by, it might break the fungus. Maybe a moose rubs its shoulder against the tree and squishes a conch into a few pieces. Maybe a fungus-eating predator comes and bites into an agaric mushroom, ripping up the main mycelia and throwing bits of it around. In either case, these little chunks of mycelia that get thrown around are still composed of living hyphaea. If they land somewhere wet, near something that they can eat for nutrients, those little fragments will survive and live on as a clone of its so-called parent. In contrast, some fungi have qualities that allow them to control, or actively or passively engage in a process of fragmentation. Lichen are a great example of this. If you just listened to the last episode, you know that I talked about lichen for quite a while. You should also know that lichen is a symbiote of fungi and algae, or cyanobacteria. The most typical way for a lichen to fragment and reproduce asexually is by simply drying out, and then experiencing some kind of mechanical disturbance, like the wind, a falling rock, the wayward hoof of a grazing mammal. You get the idea. They're hardy enough to not only survive the desiccation, but to become brittle, so that any kind of damage or contact is almost guaranteed to flake them off. And this guarantees that they spread out, and that they maximize their chances for at least one of these little bits or chunks finding somewhere fertile and growing from that, and having the genome survive and persist through time. However, lichens sometimes create specialized structures that are intended specifically, obviously, for fragmentation. In some species of lichen, these are called IDIA. The IDIA are long, thin, branching projections that come out of the thallus, or the main body of the lichen. The IDIA are quick to dry out and break off, and that's their intended purpose. The IDIA possesses hyphaea from the fungal symbiotes, and it possesses the photobiont, or the photosynthetic algae or cyanobacteria, that grow inside the lichen. Another specialized structure is called a cerealia. These are like little baskets, or cups, or bowls, and they hold a few globular structures called ceridia, so the cerealia holds the ceridia. These ceridia are just bundles of algae cells, or cyanobacteria cells, all wrapped up and tied together in hyphaea. The cerealia will open up and expose them to the wind or the environment, to rain or something. And the ceridia will get blown away or washed away, and they'll land somewhere and then grow into new mycelia clones. Single-celled fungi, called yeasts, are known to reproduce asexually through a process called budding. The yeast cell typically has a smooth, spheroid shape, and when it begins to bud, a little bubble begins growing on its side. This small bubble is the daughter cell, and it inflates like a balloon being slowly filled with water. During this little period of growth, this swelling, the parent cell's nucleus will duplicate, and the duplicate nucleus will be positioned within the daughter cell, as well as duplicates of necessary organelles and ribosomes and all of that good stuff. Once the budding cell has gone through enough of its own growing process, it'll detach from its parent yeast cell and live the rest of its life independently. An interesting detail here is that the daughter cell created through this budding process is generally smaller than the parent cell. Now, this isn't the case when a yeast divides by binary fission, which is another method that some yeasts use for asexual reproduction. Fission is a process by which a single-celled organism reproduces to create two identical daughter cells, and it's not limited to fungi. In fact, relatively few fungi actually reproduce this way. This is largely a technique that's used by bacteria and the mitochondria within a eukaryotic cell. During this process of binary fission, the fungal cell begins to copy its DNA, and then its organelles and everything else that it needs. While all of this is happening, the cell's total size is growing, and its nutrient demands are relatively high. It's got to feed all this growth, and all this DNA synthesis, and protein synthesis, and membrane synthesis. The cytoplasm bulges outwards until the cell is nearly twice its original size. A cleft appears in the middle of the membrane, and this cleft will begin to cut through the cellular mass until it reaches the membrane on the other side. At this point, the membranes meet up, and the cytoplasm is pinched off as the two daughter cells split apart. Asexual reproduction in larger multicellular fungi is naturally a little more complicated. This kind of regular asexual reproduction occurs during the vegetative anamorphic growth phase. In this phase, the fungus just grows like a mold. The hyphaea will creep around the substrate, looking for detritus to cling to and dissolve. In general, the mycelia spreads out as this amorphous blob. In the first episode of this series, I discussed the structure and the nature of the hyphaea. I believe I also made some references to the specialized growth structures that branch out of the hyphaea to serve some particular purpose. I didn't go into much detail then when I first mentioned it, because many of these structures are called conidiophores, and they're reproductive in nature. These conidiophores are structures that produce and release spores, which then float away to become new clone mycelia. Before I can get into that, I have to explain some of the evolutionary background, so you can get the full picture. There's a long evolutionary history to the fungal kingdom, but many of the earlier species in the kingdom have gone extinct, and the fossil record is frustratingly sparse on fungal fossils. It's because they have very soft bodies that are very quick to decompose, and so fossilization of a fungus is really rare. Most species of extant fungi are in a group called the Dicaria, which are fungi whose cells and spores have no flagella. They can use the wind to get their spores around. They don't necessarily need a flagella to help them swim through the water. This has also helped them adapt to harsher and drier environments. This subkingdom Dicaria has two major divisions, the Ascomycota, or the Ascomycetes, and the Basidiomycota, or the Basidiomycetes. Each of them are so named because of the structures of their spore-producing reproductive bits. In both the Ascomycota and the Basidiomycota, fungi predominantly use asexual reproduction to perpetuate their lineages. The hyphaea will split and branch, and the branching bits will develop a specialized tip. It makes a hyphaea fiber a few cells long, and then the cell at the tip divides, but doesn't extend in length. Instead, the newest cells divide laterally, and they all bunch up at the tip, all of them poking forward like hairs on a round paintbrush, or hairs on a broom. These cells might divide lengthwise once or twice more, and this would create one or two layers of smaller cells, each of them dividing again on top of their original cell. This first division you can think of kind of like your arm splitting into fingers. And this second division is like your fingertips splitting into smaller fingers. And if there was a third division, it would be like those little fingers, each splitting into 5 smaller little fingers. The purpose behind this is to amplify the number of spores that the conidiophore can produce. So to kind of help you visualize this, imagine that your arm in your hand is like a conidiophore. Your arm is a chain of single cells. It's the little hyphaeal branch that first came off of the original hyphaea. And your fingers are the long, thin cells that are bundled up at the ends of the hyphaea fiber. Now, out of your fingertips, the spores that get produced are released. So if you have 5 fingers, then you have 5 fingertips. And logically, you can release 5 spores at a time. But what if each of your fingertips was then split into 5 smaller fingers, and each of those could also produce spores at their tips? Well, now you can produce 25 spores at a time. It's an exponential increase in spore production. This is basically how the fungal conidiophores work, to produce spores for asexual reproduction. There's a wide range of ways the fungal cells produce and release these spores, and as I describe them, remember that these are single cells splitting off from the tip of a long, narrow cell, which is itself one among many branching out of the end of a single conidiophore. And funguses will have thousands, maybe millions of these conidiophores all up and running at once. So there are several blastic methods, which means the fungi uses a precursor cell to generate the spores. One such blastic method is called blastic analytic growth, where a spore will form one at a time, and then it detaches. And as it detaches, it leaves behind a scar that's shaped kind of like a ring. Every subsequent cell that gets produced, it gets produced by the membrane extending out from that same spot, and it leaves from that same spot, and it leaves scars again, and this just produces rings of scars within scars as more and more spores are produced. And so it's kind of like a tree in a way. You cut down a tree, you count its rings, you can tell how old it is. With this, hypothetically, if you were to count the rings of scars, you could tell how many spores that little bit of the conidiophore has produced. There's another method called blastic phylytic, which is a little more focused. Instead of just having the spores bud off of the end of the conidiophore, the spores have to pass through a secondary cell called a phyllide. The phyllide is a tubular cell, so it's like a little passageway or a corridor for the spore. The cell is like the barrel of a gun, and the spore is like the bullet. A third blastic method is called synchronous blastic spore formation, where multiple spores grow simultaneously from a single point at the tip of the conidiofore. Their growth is generally synchronized. They all grow at the same time, and they all bud off at the same time as a group. Then there's blastic acropoidal and blastic sympodial growth forms, which both involve a continual budding of new spores in a way that produces a literal chain of spores coming off of the end of the conidiofore. In acropoidal spore formation, the spores will keep forming at the far tip, so that the youngest spore is the one farthest out. In sympodial spores, the direction of the growth is reversed. The first spore is formed, and instead of the second form growing on the tip of that one, and going out that way, that's acropoidal, in this way, in sympodial, the second spore will form between the first and the conidiophore itself. This continues. The third spore to be produced is generated between the second and the conidiophore. The fourth is generated between the third and the conidiophore, and so on and so forth. This makes a chain of spores where the oldest spore is the one that's farthest out on the chain. There's a method called blastic retrogressive formation, which is where the spores are born when a wall is created shortly before the tip of the conidiophore. This walled-off area, this little tip section, will break off to become the spore. This will happen repeatedly, which shortens the total length of the conidiophore. Basically, the conidiophore will grow to its full length, and then as it produces these little segments and segments itself off, it'll dissolve into spores. Now, there are quite a few other cellular patterns for spore production, but I won't bore you with the details. The point is, as the hyphae are growing and the mycelium is expanding in this vegetative anamorphic growth form, some of the hyphae produce little branching conidiophores, and these produce spores. It's like a constantly active spore factory, always throwing out little spores to maximize the chances that the fungus can reproduce itself in as many places as possible, as often as possible. Both Ascomycots and Basidiomycots reproduce asexually in this anamorphic growth form. Some of these species in these groups are among the so-called imperfect fungi, as they never leave the anamorphic growth form. Their entire lives, their entire growth colonies and reproductive cycles are just asexual in this anamorphic growth form. They never really grow as anything more than a asexually reproducing mold. However, both groups also have many species that move beyond the anamorphic stage and into the teleomorphic stage. I suppose now is a good time to briefly go over the typical life cycle of a fungus. I've just explored the basics of the anamorphic stage, where the fungus grows kind of like a mold. Its hyphae extend outward, they creep along looking for food, and the whole time it's just producing conidiophores that release spores for asexual reproduction. However, there are some fungus in these groups that reproduce sexually, and this is how they get into that teleomorphic growth form, is they'll find another fungus with a compatible mating type, they'll meet and they'll fuse their membranes through plasmogamy, and they'll reproduce that way. It's a really weird process that I'll get into in a few minutes. These kind of sexually reproducing funguses are defined by what's called a mating type. A mating type is kind of like male and female in animals, except not really. There are thousands, potentially thousands of different mating types in the fungal kingdom, instead of just the two that exist in the animal kingdom. These mating types also have more subtle differences. Where animals can be differentiated by different genitals and sexual dimorphisms, the differences in fungal mating types exist on a molecular level. It's specific genes, encoded enzymes, signaling proteins, regulatory pathways, pheromones, that kind of thing. There may be many fungal mating types, but only two individuals can successfully reproduce at once. And during this sexual pairing, one fungal partner can potentially act as a male, and the other will potentially act as a female. What this means is that, with respect to the parent nuclei, especially in some species of Basidiomycots, one parent fungi will keep their nuclei, and the other parent is said to donate their nuclei into the offspring hyphaea. The one keeping their nuclei is considered the female, and the one donating their nuclei is considered the male. In a more general sense, less specific to the Basidiomycetes, some fungi are known to sexually replicate with basic gametes that they release, and these gametes will meet and fuse and fertilize. In these cases, there are larger gametes that tend to be more immobile, more stable, and as such, they're called female gametes because of their similarities to eggs. And then on the other hand, you have other gametes that are smaller and much more mobile, like sperm, and so these are just generally called male gametes. This chemically abstract male-female dichotomy allows for a degree of sexual selection within the fungus kingdom. For example, multiple males can compete with each other vigorously to mate with a single female. And in addition to this, the female has the potential to express a very strong preference, or a choosiness, for one male nuclei over another. This choosiness is based on the same principles as the initial attraction of the potential fungal mate. Fungi don't have eyes or ears, so they can't really see a mate, and they don't have mouths to make noises to call for mates, and they don't have ears to hear it in the first place. But they do have the ability to excrete chemicals and to pick up chemicals because of their high surface area, and this is something that fungi do very well. In the case of reproduction, they secrete pheromones, which are typically effective over short distances. They allow an individual mycelia to detect and assess the quality of another mycelia in close proximity, to recognize its mating type, and to maybe get some input of its health, of its fitness. On the cellular scale, pheromones describe the quality of the nuclei. Some nuclei with perceived poor quality can be rejected by the female fungi because she perceives it to have a weak pheromone signal. It's that weak pheromone signal that's associated with that perceived poor quality, that low fitness. Some pheromones are water-soluble, where they can be released into the water by one fungus and detected by another. The general trend is that mate choice typically goes to the individual, or the gamete, with the highest concentration or the strongest attractive pheromones. More pheromones simply induces a stronger preferential effect, and directly increases mating success. When two compatible mating types meet in physical proximity, they can extend their hyphaea and initiate the homing behavior that begins an anastomosis, or a conjoining of two different filaments of hyphaea. The anastomosis that forms literally fuses the parent fungus together. They literally co-create new cells in this symbiotic limb, this symbiotic filament of hyphaea. They merge their membranes, and they mix their cytoplasm and all of their organelles together in this symbiotic tissue. This process of the merging of the membranes is called plasmogamy. The membranes and the cytoplasm come together to compose the new cells, but the compatible haploid genomes aren't combined analogously. They actually aren't joined together to make diploid cells. Not yet. Instead, they temporarily form dikaryotic cells, which are cells that possess two distinct haploid nuclei. They're not bound together like they would be in a condensed diploid nucleus. At this stage in the life cycle, they're called pronuclei, and this kind of foreshadows their future merging. In some species of Basidiomycots, this dikaryotic prekaryogamy stage is characterized by structures called clamp connections. Within the very tip of this growing dikaryotic hyphaea, cell replication is happening. It's happening at the very tip. Each time the cell at the tip replicates itself, it extends the hyphaea, and it also has to replicate both of its nuclei. Concurrent with this DNA replication is the emergence of a little protrusion coming out of the side of the hyphaea cell. It's called a clamp connection. This protrusion begins to form an arc, or a hook structure, and as it forms and it extends, it sucks up one of the 4 nuclei that now exist in the cell. As this nuclei moves down the growing tube, one nuclei from the other pair will migrate to the rear end of the cell. A septum is formed in the middle of the cell, and it's between each copy of the genomes. This also forms a physical division between the two new cells. Now the clamp tube is still growing. It reaches back into the second cell of the two new cells and deposits the nuclei that it was carrying. Now both cells have one copy of each nuclei, which preserves the dikaryotic nature of the hyphaea as the cells replicate. In these species of basidiomycots, this also creates long filaments of hyphaea that are lined with clamp connections. All the cells in their dikaryotic hyphaea are linked with little loops of cytoplasm and bits of membrane, like a little braid running through the cellular chain. After some period of time, these pronuclei go through a process called karyogamy, where they fuse together into a single diploid nuclei. Karyogamy begins when the pronuclei move together, in a step called pronuclear fusion, or pronuclear migration. In the mid to late stages of plasmogamy, enzyme complexes of kinesin motor proteins and long bundles of microtubules, they were all constructed. And now, in the later stages, during this pronuclear migration, these microtubules that are built around each nuclei will orient towards one another, and they work as a means for the nuclei to pull closer together. Little kinesin motor proteins crawling along the length of the microtubules will pull them past one another, kind of like filaments in a muscle cell. After pronuclear migration is complete, the early steps of true karyogamy, they begin, and they involve the nuclear double envelopes merging together. First the outer layer, and then the inner layer. The spindle poles, which are these microtubule complexes, they'll also fuse together. The nuclei themselves will meet, and through stabilizing enzymatic reactions, they're able to be merged together and fused, in much the same way that chromosomes are swapped and merged and organized during various stages of meiosis or fertilization. At the end of the process, the replicating cells in the new hyphaea are diploids with a genome similar to, yet distinct from, the genomes of either parent. In a nutshell, this new diploid individual has diploid cells, and through meiosis, they'll shuffle up the traits between these separate parent nuclei, and though they'll produce haploid spores, these haploid spores will have a randomized, unique genome that's it's unique from both their parent and their grandparent's genome. These haploid spores then get released, where they can land, germinate, and create more haploid mycelia, and the reproductive cycle begins all over again in the anamorphic stage. The haploid spores produced by this diploid mycelia are grown in specific fungal cellular structures, which kind of depends on the type of fungus. The spore-producing cellular structures are embedded into a relatively thick fungal tissue called a fruiting body. The fruiting body is the large, dense, macroscopic teleomorphic growth form of the fungus, which you might recognize as a mushroom, or a conch, or a puffball, or one of literally hundreds of other types. When I say that they're large and dense, I really mean it in relation to the individual hyphaea. If you look at a mold, it's a thin, soft carpet of hyphaea, kind of just growing peacefully on a substrate. In comparison, a fruiting body is much denser, much stiffer, and it's much more resistant to physical trauma. I mean, they're still relatively soft, and they can be eaten by most organisms, but mold itself is pretty much totally defenseless from mechanical damage. Fruiting bodies are macroscopic. They can be seen with the naked eye, whereas individual hyphaea are microscopic. You have to use a microscope to see them, and they can only be sort of seen with the naked eye when they group together to make rhizomorphs, or if they form bundles that, to us, look like little hairs coming off of them. These funguses are teleomorphic because, well, this is the teleomorphic growth form, and they experience it. They go through it in their regular reproductive cycle, or their life cycle. The fruiting bodies are centralized, spore-producing structures created for the mass release of these haploid spores with these randomized genomes. Now, this is a kind of a neat little evolutionary trick. If most basal funguses reproduce asexually, then they might suffer more from problems associated with repeated cloning. The lineages will begin to accumulate mutations, and this will create unfit individuals. Furthermore, populations of clones are at risk of diseases and alterations to their environment, because the population lacks the variety in genotypes and phenotypes to survive these stressors. Because of this, being able to reproduce sexually is a particularly valuable thing, as it mixes up genes and creates unique genetic combinations. It creates genetic diversity, which promotes the health of the individual and the health of the population. You can clearly see the evolutionary value here. The fungus saw it too, as they evolved a diploid growth form, this fruiting body that forms from that symbiotic dikaryotic hyphaea. This fruiting body would produce a vibrant and massive explosion of spores with new genetic material. And even though this process will create, little haploids with just single copies of their genomes, and these will only be able to create clones of themselves going forward until they go into their own teleomorphic phase, they'll have genomes that are distinct from either parent. So where does this process of karyogamy typically occur? Which cells are affected? These are good questions, because it happens specifically in those specialized spore-producing structures that I've mentioned so many times now. I've also mentioned two major groups of fungi, the Ascomycots, or the Ascomycetes, and the Basidiomycots, or the Basidiomycetes. The names of these two groups both denote the structures that they use to produce spores in this teleomorphic form. For example, the Ascomycetes fungi have a reproductive structure called an ascus, which is kind of like a thin tube. This ascus will experience karyogamy, and its diploid cells will then undergo meiosis to create haploid spores, called ascospores. It'll typically produce about 8 of them. In some species of Ascomycots, they'll produce about 2 spores per ascus, or 4, or a multiple of 4. Although 8 spores per ascus seems to be the most common in the fungal species. Now, on the other hand, the Basidiomycetes, the Basidiomycots, use a different structure for this, but the general idea is the same. Even the naming conventions are the same. The Basidiomycetes have reproductive structures called basidia, which are shaped like a club instead of a tube. The basidia goes through karyogamy to become diploid, and then its cells produce 4 haploid spores through meiosis, and these are called basidiospores. Some species of Basidiomycetes can produce 2 spores per basidia. Others can produce 8. But 4 basidiospores seems to be the general norm. The basidia and the asci are specifically grown in a layer of tissue called the hymenium. This tissue layer is typically external, where it lines the body of the sporocarp, or the fruiting body, in such a way that the spores can be easily released into the external environment. For example, your typical agaric mushroom has gills on the underside of it. These gills have hymenium layers that produce and release the spores into the air. They fall off the sides of the gills and land below the mushroom. In bolete mushrooms, instead of gills, the hymenium actually lines the inside of vertical tubes. These tubes run down the interior of the sporocarp, where they end in little pores along its bottom side. The spores produced in these tubes are released, and they simply use gravity to tumble out of the tube. Once free of the parent fruiting body, the spores can be washed away in the rain, or blown away in the wind, or picked up inadvertently by a nearby animal. In cap fungi, the hymenium is on the inner concave surface of the sporocarp, where the spores can be blown out with wind, or scraped out by an animal. Most commonly, the cap fungi will spread their spores with rain. Water droplets from rain, or from water just falling off of a tree or something that happens to be above the fungus, these water droplets will fall into their cup or their bowl-shaped depressions, and the splash will propel a number of spores out of the hymenium tissue and into the nearby environment. In stinkhorns and puffballs, the hymenium is grown on an internal surface, integrated with other tissues so as to create a bubble or a cavity that's pressurized with air or gel or some kind of fluid. It's designed to be burst open. The force of this blasting open is enough to suction out or propel the spores, throwing them all over the place like shrapnel. There's even a few species of fungus where their spores are enclosed in flask-shaped containers with small little openings at the top, very small, and if a water droplet, like rain, happens to land on it and get inside, the water will react with the internal biochemistry to shoot a single spore out of the little flask structure, one at a time. This chemical acceleration is tremendous, something like 10,000 Gs. However, the spore is kind of small, and the chemical force itself operates on a relatively small scale, and so this massive acceleration only shoots the spore about, less than 1/5 of an inch. But that's enough. That's enough to get it to tumble out of the parent fungus and get out somewhere in the environment. In parasitic fungi, the spores or the mycelia invade a host, and they use its body as a staging ground for the production of more fruiting bodies. This is the case for the infamous Ophiocordyceps unilateralis, which invades the body of an ant and turns it into a zombie. There are a whole lot of other species that are kind of parasitic in this way, like zombie funguses that infect some kind of arthropod, and they go beyond the species unilateralis. They largely belong to this genus Cordyceps. So Ophiocordyceps unilateralis has spores that can latch onto the exoskeleton of an ant, and they can exert mechanical forces through chemical means to puncture the exoskeleton and break through it to reach the soft tissue inside. And then they begin to chemically take over its body, to hijack the body from the control of the little ant mind. The ant's mind, or its consciousness, if you will, whatever awareness the ant has, it becomes a prisoner in its own flesh. The first behavioral modifications include irregular spasms that are intended to destabilize the ant. If it's crawling on a branch or on a plant, the fungus wants the ant on the forest floor, so it induces these spasms that make it kind of lose its footing and fall to the ground. Once on the forest floor, the fungus begins to exert more control, and it induces the ant to find a vascular plant, and to crawl up its stem and out onto a leaf. Then, the ant is made to bite extremely hard on the central leaf vein, so as to position its body in the right spot to produce the fruiting body. This spot, this specific point geographically in relation to the ecosystem, is oddly specific. The fungus aims for areas on the plant about 22 to 28 centimeters above the ground, where it's very humid and generally warm, around 20 to 30 degrees Celsius. After the ant has been used to position the fungus will finally kill the ant, and then it will begin to earnestly consume its soft flesh for nutrients. Hyphaea will grow to reinforce the insect's exoskeleton as a kind of protective shell, and in the course of 4 to 10 days, the fruiting body breaks out of the ant's head. It comes out as one or more long, stiff tentacles, grasping and growing through the air. These structures contain hymenium tissue, which produces more spores that can then be released to potentially infect more ants. These fruiting bodies look different depending on the species or subspecies of Cordyceps. Some are white shafts with spherical red caps. Others stay shaped like tentacles, which grow brown or white, gray or tan or yellow. Some grow shaped like clubs or flasks. This wide variety isn't limited to just Ophiocordyceps. There's a huge variety of morphology across fungal fruiting bodies. Some are brightly colored, others are dark or dull. Some are simple globular shapes, or typical agaric shapes, while others have an exotic, alien appearance that can hardly be described. Furthermore, fungus are hugely diverse in their chemical properties. Some are safe to eat, and they taste delicious, and some are safe to eat, but they taste disgusting, while some are dangerously poisonous and they can kill a human. Others still are consciousness-altering, in ways that many describe as spiritual. This wide diversity in morphology and biochemistry are testaments to the ancient lineage of fungus, which has gone through more than 1 billion years of evolution. I'll be exploring fungal evolution in great detail in the next episode, so if you've been enjoying this series so far, be sure to stick around and check it out. If you really enjoy my content, if you like all of these series, then please subscribe to my channel, give my stuff some likes, and share your favorite episodes with your friends. And as always, thanks for listening.
Would you like to support the BioLogic podcast? It's super easy. When you open a new episode, press the like button or share it with your friends. If you aren't subscribed, you should hit the subscribe button so you can enjoy a new episode every week. You can also peruse our official store, which has a ton of cool stuff like hand-designed t-shirts, hoodies, and stickers. All the links you need are in the description section below.